Welcome to the presentation. Today we will give you a quick overview of our scavenging technologies, in particular the E-PAK technology. Nowadays there is a lot of pressure on the drug discovery process where we're asking a lot of innovation to be done in a very small amount of time. And we could see that today with COVID-19, where a lot of the pharmaceutical industry had a lot of pressure to get a vaccine as quick as possible. To give you a quick summary of how that goes, the research phase takes usually between 3 to 5 years, where you get the target identification, followed by the drug molecule that will heal the problem, then optimizing the synthesis of this molecule, followed by different preclinical and clinical tests to make sure that the drug is efficient, followed by the late phase. My name is William Sommer. I have a PhD in organic chemistry, and I'm a product manager at Cycle. And today I will talk about E-PAK a bit more in details. I've been working in the chemical industry for about 15 years. So how does scavenging work? Well, you have an API, your API, with an impurity that you want to remove. You put in your scavenger for a certain amount, you stir it together, and then doing a filtration where you remove your scavenger with the impurity and you get your API at a higher purity. You have different parts where you can put your scavenger. You have the choice. You can put it right after the catalytic reaction, or you can put it before the last step of the reaction. Usually, if you put it at the last step, you will be required to put a bit more scavengers because you will have more impurities that could have gone within the different steps of your reaction. So how do you do that? Well, the first step is to choose the right scavenger. Then once you find it, you can optimize the lab scale and then the scale-up optimization. So to find the right scavenger, you can use a table that is available from us on our website or on different publication. And you can see in the table that we have almost any kind of scavengers to any kind of metals. So the first step is to pick the different scavengers that you want to screen, find the one that works the best, and then move on to the lab scale optimization. Once you did your screening procedure, you need to optimize the different parameters to make sure that you get the more efficient scavenging. So you can optimize the reactor by looking at the size of batch, or if you're using a fixed bed. You can vary the number of equivalents that you can use. You can do subsequent treatments, vary the reaction time, the temperature, the type of solvent you're using, or how fast you're mixing the reaction. And you can see here on the graph, is an example of a removal of palladium acetate with a CDMA tile in DMF. And once you increase the number of equivalents and the temperature, you can see that you can get almost all of it removed very quickly within 5 minutes when you put 4 equivalents at 80 degrees. So I mentioned that you have different type of reactor you can use. So you can use a batch mode, so more conventional, where you can see the reactor here and it steers, or you can use what we call a fixed bed or flow, where you have your reactor with your reaction, and then the reaction can go into a housing that you can see here on the picture there, where you have your scavenger, in this case, the E-PAK cartridge that I will describe in a little bit, and then it can come back into your reactor with your purified solution. So what is E-PAK technology? This is all your well-known CDMA silica, functionalized silica, and activated carbon in a cartridge form. So it ensures a rapid, simple, and reliable technology, making it easy to handle, to clean up, and to dispose of. And you don't have to do any filtration steps, since your solvents with your API goes through the cartridge where the scavenging is occurring. Furthermore, it has a very large absorbent capacity in a very small footprint area compared to the other technologies that are out there. It increases the product recovery, it reduces the solvent requirements that you might have, and it's safe to use with all common solvents. It has a high absorption capacity, and the flow rate is radial, meaning it goes from out to in from the side, allowing you to not have to put too much pressure to get your solvent through the cartridge. Since it is a cartridge, it allows you to have multi-pass functionality, meaning that you can cycle through your solvent several times through the cartridge, and you can put different cartridges or several of the same cartridges, increasing your capacity. So you can use it in different methods. The most common one is a single pass, where you have your solution going through the cartridge and then going into another vial purified. You can have recirculation, where you have your solution going through the cartridges several times. You can have multiple sequential pass, where it goes from one housing to the next one and then go back to the other way, going through the cartridge. And you can have series pass, where you can have multiple cartridges, where your solution can actually go through different type of cartridges, removing the impurities. We have a wide variety of sorbents for our E-PAK cartridges. I mentioned earlier, we have activated carbon. We have 4 types of activated carbon, and we have the most common Metes product in our E-PAK technology. So we have the thiol, the GMT, the AMPA, the imidazole, the diamine, triamine, TACOH, TACONA, the cyano and the fluorosil, and the propyl sulfonic acid. And of course, the different E-PAK sorbents have different affinity for different metals. So depending on what type of metals you want to remove, you can use either one of them. So once you figured out which scavenger you want to use, you can start to proceed with the lab scale optimization. So in the case of the E-PAK, it's really a continuous flow reactor, where you can put your lab scale cartridge, which is a smaller cartridge, which is easy to put in your housing. You can see the picture here below. We have 3 types of cartridges: the 5x1, 5x10, 5x25. Those are the size of the cartridges. Depending on the size of the cartridges, you're able to use different flow to put through it. And of course, the size of the cartridges will dictate how much carbon or silica you have in the cartridge. So when you do your calculation on how many equivalents you need to have to remove your impurity, that's what will determine the size of the cartridge you will use. Once you figure out the lab scale optimization, you can move on to the pilot and commercial scale optimization. In the case of the E-PAK, the beauty of this technology is that you can actually use all the parameters that you determine with the lab scale, and using a coefficient be able to use and get the same result with the bigger scale. So for the pilot scale and commercial scales, we can go from 10 liter up to 10,000 liters. And you can see we have those cartridges have a bigger diameter, 16.5, and they go from 12.5 centimeters all the way to 1 meter long. Once again, depending on the size of the cartridge, you will have different flow range you can use, and the quantity of the silica and of carbon will vary depending on the size of the cartridge. So you also have multi-cartridge housing, where you can put several times the same cartridge, or you can put different cartridges to remove different type of products. So we have a 3-round, a 7-round, and a 12-round. So as I mentioned before, the beauty of the E-PAK technology is that you can use the parameters that you've determined for the lab's efficiency, or you can use the same parameters with a scale-up factor to use for your pilot scale or to your commercial scale. So that means that you can extrapolate the scavenger to impurity ratio, the residence time, the temperature, the solvent, and the contaminant and compound levels that you have. So in the next table, I will show you those parameters and what they mean. So we're going to go a bit slower on this table because we have important parameters to talk about. You can see the first line, you have the different size cartridges that are available from 5 by 1 centimeters all the way to 16.5 by 100 centimeters. You have your scale-up factors, those so that the factor you will use to multiply by for the cartridge that you use. So if you use a 5 by 1, the scale-up factor would be 10. When you use a 5 by 10, all the way up to 16.5 by 100, you can see that the scale factor is 875. So scale factor is really the mass of silica that is in your cartridge, and that's what determines how much silica you will use from your 1 centimeter all the way to your 100 centimeters. The bed volume is the total volume of your cartridge, and it counts the calculation also take into account the volume of the hole. You have the recommended flow rate. So you can see we have for the 5 by 1, 7.5 milliliters per minute, all the way to 7.6 liters per minute for the 16.5 by 100 centimeters. So the recommended flow rate is determined by the residence time, and what we call the residence time is how long your product stays within the housing and how long it stays in contact with the cartridge. So the most efficient time that we've experienced so far is 2.5 minutes. So the 7.5 milliliters per minute correspond to 2.5 minutes residence time of the product within the housing. The flow rate range is the range that you can use for each size cartridge. So you can see it goes from 1 to 20 milliliters per minute for the smallest one, all the way to a liter to 20 liters for the biggest one. The conditioning is the amount of solvents that you need to go to put through your housing to get it prepared. And experimentally, it's 3 times the volume of the housing. And you can see, so for the smallest one, 150 milliliters that you need to put as solvent, all the way to 70 liters for this biggest one. So schematically, how does it work? You have the solution with the impurities that you want to remove in your reactor. You will hook up the housing, in this case, in a single cartridge housing, and then it will be hooked up to the final reactor that will have the clean solution in it. And finally, it's important to note that CMATS and E-PAK can help you meet the new risk guideline from the ICS that was published in December 2014. And is also FDA compliant. It can be used with food and drug application. And Silicycle can help you with different regulatory filings that you might have. And we can also do some stability studies for you. Let me tell you a little bit about Silicycle. We're headquartered in Quebec, Canada. We have offices all around the world in the US, Europe, India, and China. And we serve over 100 countries. For more information, check our website at silicycle.com, where you can find our application note, our case studies, and all of our products. If you have any questions, do not hesitate to email us at support@silicycle.com. And don't forget to check our YouTube channel, where you can find all of our videos. Thank you for listening and have a great day.